I will invite Mrs. Balu to come on and start asking you some questions. We have a lot of very thoughtful questions to present to you. Mrs. Balu.
Yes, yes, Teresa. Thank you, Darin, for a very informative session. We have a lot of questions coming up. Let's see if we have time in the half an hour or if we have a few minutes fast. Otherwise, you have to type in the answers and then we can put it on the blog like we mentioned before.
Right.
So, yeah, they wanted to know, the first question is, what is the percentage you will give for genetics as a flavor precursor?
Um, well, so that's okay. So you really start off with hard questions, right? So you can have the best genetics, the best flavor potential, but if the processing is not appropriate or does not really express that potential, then that genetic potential is unrealized, right? Um, so I I I don't think that the question of saying what is the percentage of genetics for as a flavor precursor, I think it's really what is the potential that exists in that variety and how you express it. So it goes back to it goes back to understanding the the genetic ancestry of the variety, and by looking at different fermentation approaches, what is the best fermentation approach that expresses a flavor potential that you think represents the best expression for that variety. And there's also the overarching notion of terroir, where we did a study where we took the same varieties that were grown in Trinidad, Ecuador, Venezuela, and Papua New Guinea. So this is like 4 different parts of the world. And they were grown and processed in those countries, and we made them into liquor, and they tasted totally different. So there was an impact of terroir, the combination of the growing environment, the processing microorganisms. So we are now in our own process trying to deconstruct elements of the exogenous environment. So this is why, as I mentioned, that we are engaging in this omics study to understand the microfloral contribution from the environment. What is the population of the microorganisms throughout fermentation, how it progresses, and what is the contribution of their metabolics of the metabolic products from the microorganisms to the fermentation and flavor development.
Okay, the next question is there any relation between the altitude and flavor?
Altitude and flavor. I think what happens, the higher up you go, you get a colder ambient environment, and fermentation occurs in the context of the environment in which it happens in. So if you are higher up and you have a cold environment, then you would have a lower optimal temperature, or a lower temperature that may be suboptimal for the microorganisms. So they might move at a slower rate or be less active. So it's not really the altitude affecting flavor, but the altitude affecting the environmental conditions that impact on the microbial succession. So you need to look at the temperature and the relative humidity of the environment to understand that. But understand that if you have lower temperatures, you have a lower incidence of microbial activity because it's cooler.
Okay, the next one is, they're saying in coffee, in higher altitudes, the pod is stuck to the stalk longer, to the plant. It has more time to absorb the sugars. Is it true in?
Okay, so the pod ripening process, cocoa is non-climacteric, right? So it's not like a banana. And they found that the ambient temperatures during pod maturity affects the rate of ripening because it affects the enzymatic systems that occur during the ripening process. And in terms of both internal, in terms of the change of the starches and sugars, as well as the enzymes that control the coloration of the pod. So you might tend to have a bit longer time on the tree where the pod stays in an optimal condition. So yes, you could have some more accumulation of sugars occurring, but I haven't tested it myself. I'm just answering pragmatically. But also at higher altitudes and cooler temperatures, it has an impact on the cocoa butter hardness because they found in there was a study published by Butler et al. In the 1980s where the cocoa grown in the south of Brazil that was actually cooler had a softer cocoa butter than cocoa grown in the north of Brazil that's a bit hotter. So the hotter area, so the same way cocoa from Malaysia where it's hotter tends to have harder butter. So it affects the cocoa butter hardness as well.
Thank you. What's the next one? They're asking if the cocoa is growing in dry seasons, how do you calculate the water needs of the trees with drip irrigation? And it's irrigation related.
Yeah, so that's actually a plant physiology question that I can't answer that question. What I can say is that drawing on the experience from Ecuador, a lot of their large plantations and new plantations were moved to areas that were drier and they had to bring water to these fields, and they invested a significant amount of money towards irrigation systems, the drip irrigation and so on. The benefit of that was that you have a lower incidence of fungal diseases because you have drier conditions, but you also, by being able to exercise access to water to the plant, you could actually also incorporate fertigation or the application of fertilizer and also affect the flowering cycle of the plant because flowering is linked to water stress. So you can actually modulate that with fertigation to have production all year round. With respect to the actual needs of the plant, I think it is on the physiological state of the plant that the physiologists would have worked out. So I can't answer that, sorry. So that specific part of the question.
The next one is, they want to know, is there any good bricks level to harvest the parts?
Sorry, if there's any what?
Bricks level, the sugar level. Can you calculate the sugar level or just the sugar level for harvesting the parts? What's a good,
So what you can do, you can actually measure the sugar level in degrees Brix of the pulp before going into fermentation. And this is something that we're actually now starting to do. And we're tracking to see how that sugar level changes during the season and across crop years. So we are still actively engaged in that process to understand this better and understand the impact on optimal fermentation time. But it is possible to measure it. You have handheld Brix refractometers as well as benchtop models that you can take a subsampling of the pulp and measured Brix. What's also more important in addition to Brix is the pulp moisture content, right? And the amount of pulp. So it's possible to work out the pulp index for a variety. The amount of beans that gives 1 kilogram of pulp, you can extrapolate it based on a small sample. And that's useful in understanding the pulp characteristics of a variety and the possible impact on fermentation quality.
Actually, they're asking about the harvesting the pod. To harvest the pod, do they have to test the bricks in the pod?
No. Well, you can't to test the bricks in the pod. You would have had to harvest the pod already. So the question is more linked to how do you determine optimal pod maturity. So there are a number of ways, some easier than others. One is looking at the change in coloration of the pod, of course. So you have green pods turn yellow, red pods turn orange, but you have so much mixed in cocoa that becomes blurred. I know, so the most accurate way, of course, is to count the days after pollination would have occurred, right? Because you know, a cocoa takes between 5 to 6 months to mature on the tree from successful pollination, right? Um, so that's the most accurate way. At the moment, the optimal maturity of a pod for harvesting is assessed pragmatically based on experience, based on coloration, a change in color, which could be affected by nighttime temperatures. Um, it's something that's developed with experience. Some varieties have very subtle changes. For example, ICS 95 is one variety where for me, even after working in cocoa for 25 years, I look at ICS 95 and I'm thinking, is it really mature or not? It looks, so some varieties are easier to understand when it's mature, when it's fully mature and ripe for harvesting than others. So it's still a sort of pragmatic based thing. But days after pollination is the most accurate way, or days after successful pollination.
Okay. Now, somebody wants to know that a little bit more about what is aerobic fermentation and what is anaerobic fermentation. You said more in technical terms, but can you say in like a layman terms? So who doesn't have the background in
All right.
science can.
All right. So anaerobic fermentation is fermentation that occurs in the absence of oxygen. And if you look at a fresh cocoa bean with its pulp around it, it's very fleshy. And when two beans come together, the pulp forms a sort of airtight seal. When the pectinase enzymes break down the pectin in the pulp and the yeast metabolize the pulp, and the pulp breaks down, you don't have that tight seal anymore. So you have oxygen coming into that fermentation matrix. So then it becomes aerobic. So it really has to do with the presence or absence of oxygen. Anaerobic is without oxygen, aerobic is with oxygen. And yeasts can function best without oxygen.
The next one is they're saying in a coffee, they use a system with the recycled juice tank with the hose attached that goes to water bottle. So carbon dioxide comes out and oxygen doesn't get in. I don't know, I didn't understand that. Okay, let me do the next question. Okay, the sensory evaluation standards. They're saying the Eastern and North American specialty coffee already has a lot of common standards. So in your opinion, how far is the fine cocoa sector away from such a unified approach?
So that's a really nice question. So at the moment, so there is an international working group for the standardization of cocoa and flavor quality assessment. So it's the cocoastandards.org is the website. So this is an initiative that was started in 2015. I am a part of that initiative. Dolores, if she's still on the call, she is very actively involved in that initiative. It's coordinated by the alliance between SEAT and Biodiversity. There's an industry group working together with representatives from both large and small chocolate companies and research coming together to create a sort of harmonized standard for quality assessment, both physical and sensory assessment. So there are draft standards available for flavor assessment for physical and sensory assessment that are available to download and give comments on. So that activity is very well advanced, and I'm happy to be involved in that. Actually, the first work started on that. I did the first review on that back in 2015. So it's happening.
Okay. Somebody is asking, since you showed in the slide, the day 4 is good for fermentation, it is done. So should they stop the process in 4 days?
Okay, so this is where there needs to be the change away from looking at a recipe where I stop all my fermentations at 4 days or 5 days or 6 days, but understand what is what is the temperature and what is the pH profile that would facilitate that decision, because it could change. At the start of the crop, you might have longer fermentation times. So 5 days fermentation might be optimal based on the measurement of temperature and pH of tester and cotyledon. At the end, towards the end of the season, it might be down to 4 days. So it's not a rule or a recipe where we say, okay, now we stop 4 days. That changes as the season progresses. It changes. It can change according to location. So this is the big change in mindset that we have to take when we look at flavor expression and the approach towards fermentation. Fermentation is a biological process occurring under ambient conditions in a natural environment. So the rate at which the reactions occur varies according to the conditions around it and the intrinsic characteristics of the beans, that is the sugar content, etc., inoculation and so on. So each fermentation batch is a unique entity unto itself and must be treated like that. So it's only by really measuring and understanding the cues, sight, smell, and taste, along with the measurements, you can tweak it. You say it's a similar approach to if you're cooking. You use different cues to determine optimal endpoint.
Okay. The next one is they're asking, like fermenting in the wooden box. Does it, contaminate with any contamination from wooden box? If it has rusty nails, is it going to affect? So what are the things that they have to look for when they're using the wood?
Okay. If you look at wooden boxes with nails in it, with nails that are present on the inside, and beans that come into contact with those nails, you get those beans actually having black spots on them on the outside because of the acetic acid coming into contact with the metal. So ideally, all of the metallic bits that hold the box together should be on the outside. The fermentation box itself has to be handled in a certain way where you would want to clean it. Between fermentations, you would want to clean it very well at different points in time during the season. We wash our boxes out with vinegar sometimes because sometimes you have higher fermentation rates occurring on the tops and corners of the box. So when we see secondary mold growth occurring on the beans during fermentation on the tops and corners, we would then spray our boxes down with normal vinegar that you buy in the supermarket. It's a natural way to treat with that. We would scrub our boxes out with just water and a scrubbing brush. At the end of the crop, or before we start back the crop, we would pressure wash our boxes to remove any secondary mold growth, and we spray it down with vinegar. So it's a very organic process that we follow. No harsh chemicals like bleach and so on, but you can just scrub it down or power wash it and use vinegar to spray it down. Wood itself, the whole idea is to use wood that is not resinous, like pine and so on, where you have a lot of resin in it. It could impact on the flavor. You also want to have wood that doesn't splinter or break very easily. So Cedrola mexicana or cedar is used in the Caribbean because it's available. It's lightweight. Termites don't like it, and it doesn't really contribute to the flavor. I've never seen a study that shows the fermentation, the wood that's used in the fermentation box contributes to the flavor. I know if you store beans, the finished beans in different types of woods or oak casks and all that, there is the possibility to impart a flavor onto the beans. Whether or not this flavor holds on adheres very strongly to the beans to withstand roasting is the next issue or next question.
Okay, how about the temperature? Because some places it goes around 50 degrees Celsius. Is it a too high a temperature for fermentation?
Now, once it crosses 45 degrees, you're good. I've seen temperatures go up to 50, 52 degrees. So one of the things that happens in fermentation at temperatures above 45 degrees, you also have the formation of pyrazines. Pyrazines contribute the sort of nutty chocolate flavors to beans as well. So you have pyrazines formed at the elevated fermentation temperatures and during roasting as well. So 50 degrees is not too high, 51 degrees, 52 degrees is not too high. That is just what's happening with the fermentation mass. But at that point in time, we know that that's the Vmax of the microbial action. So it's going to go downhill from there. So you need to make sure that you note smell and how the beans look to end fermentation.
Is it possible to create our own flavor notes during the fermentation process?
Right. So this is from Abdul Karim. Yeah. Um,
On the dot.
yes. So this is the elephant in the room that I didn't speak about, and this has to do with modified fermentations, where people are adding different things to fermentations to introduce flavors. So Valrhona has 2 commercial products produced from double fermented beans, where they add fruit juices and so on to fermentation. I know in Ecuador they do some work. They have products for this. By and large, this is something that's happening. Sometimes it's been done by trial and error. My take on this is that yes, you can introduce flavors. The thing is for the flavors to be introduced in a way that it is stable in the bean and pleasant when the beans are roasted and converted into chocolate. Now, I know that there is also a patent called a patent by Bertus Esquez on trop mix fermentation and animal fermentation, where it speaks to those things. For me, everyone has a perspective, right? My perspective is that we should look at the diversity that exists in genetics and look at how we can optimize the fermentation process to express that genetic flavor potential.
Okay, this is regarding the genetic mapping. Are there, do you know any institutions that do the genetic mapping? And does University of West Indies, do they do the cacao genetic mapping? And if there are any fees and how are the costs calculated?
Yeah, so yes, the Cocoa Research Center, we do offer that as a service, an international service. This is Estella, I think. She can send me an email from the presentation, or send you an email and forward it to me, and I will forward it to my director, and we can handle it from there. But yes, so the Cocoa Research Center, we do in fact do genetic mapping. We will give advice on how the sampling should be done and so on for that.
Okay, and the method of fermentation can be developed, any proven study to show the method of fermentation can develop good flavor beans?
Yeah, so there's a whole range of published work on different fermentation approaches linked to flavor. Like I said, when I started, every country would have their different approach to fermentation, whether or not it's done in heaps or trays or baskets or boxes. There are a number of studies that have been done to highlight the positive and negative elements of some of those fermentation systems, and what could be the most optimal approach. For me, for final flavor cocoa, fermentation done in a fermentation vessel, a wooden box, tends to bring out the most ideal set of flavors and allows for control of the fermentation process. And there are published books on this. A search in Google Scholar will bring a whole range of references. If you just type that question into Google Scholar, you will get a range of references to give a lot more detail to that. And
Okay.
yes, we have done some work at the Cocoa Research Center in that as well. That is available from our library.
And somebody wants to know the information about the thermometer that you show in one of the slides about measuring.
Yeah, so that's available on Amazon. I can send that link. It's I think 56 US or something like that for it. We bought it on Amazon. Yeah, it's fine. It works. We use it, and it's robust enough that we've given it to farmers to use, and it survived and has come back in one piece. So we use that for our fermentation, and the stem is long enough to give you a measurement at the bottom of the box, middle, or even at the top of the box if you wish.
Okay. And then, some the proper drying storing methods. Can you store the beans for 3 years or more?
It all depends on the conditions of storage. I have seen beans stored for 19 years. I'm not saying that the beans look pretty after 19 years, but you can store beans once temperature, moisture, pest control are handled properly. You can store beans. Typically, beans are not stored for a long time in the tropics, but um, it's all about how you manage temperature, humidity, and pests.
Does grain pro packs or sacks, do they also protect the beans from flavor contamination?
Um, the grain pro bags are actually breathable, so I don't think that they will protect against flavor. But I've not tested it myself. They are not impermeable. I think one of the unless I'm wrong, but my understanding is that some of the grain pro bags are not impermeable. They allow some breathing to occur. So once you have exchange of air, you also have the possibility of introducing off flavors depending on the intensity of the off flavor, the extent of the length of duration of exposure to the off flavor, etc.
Okay. The next one is in which step of the fermentation the cocoa flavor enhanced?
In which step of the fermentation is cocoa flavor enhanced?
Yes.
So the definition of step is made what? Is made through is it by dividing fermentation in aerobic and anaerobic, or because they there is a continuum that exists, right, with the microfloral succession. So if you do not have an appropriate and adequate anaerobic phase, then you have less metabolic products to fuel the aerobic phase. So it's connected. It's an interconnected activity. So this is where we are now trying to understand the dynamics that in detail, looking at the microfloral DNA and the metabolomic products to understand the extent of the interplay within a fermentation mass. So for me, there's no one point where the fermentation, the flavor is developed. It's all a continuum that starts with good quality beans going into fermentation.
Okay. Is it possible to do vacuum sealed bags for storing the beans to avoid contamination?
Yep, yes it is. What I found when I vacuum package nibs, even sometimes, the vacuum doesn't hold tightly because it's and maybe it's because I don't have an industrial strength vacuum sealer, right? Um, you have the beans that become held tightly once the vacuum is pulled. But if those beans move because they're loose, right? If the bag is moved and that structure is broken, then you lose the tightness that would have occurred. Because I don't think you achieve full vacuum unless you have a very powerful commercial vacuum sealing system. But yes, you can store beans ideally under in a vacuum sealed system. Um, yeah, I think there's a technique called push box where they are looking at shipping beans in boxes. So versus having beans stored in bags, is boxes with beans are put in plastic bags, and I think it was vacuum sealed as well. But it's very expensive, obviously, and you can't put 67 or 68 kilos of beans in a vacuum seal bag. It might have to be smaller volumes. So it will change the logistics of handling those beans. Right. Any more questions?
A lot more questions.
A lot more questions. Okay.
Okay. They want to know from Puerto Rico. They want to know any guidance on a tree to bar traceability software.
Tree to bar traceability software. Okay. So at the Cocoa Research Center, we actually we put together a traceability system that we are rolling out that uses QR codes assigned to lots of beans from farm to fermentation to drying to warehousing. So we have that we have developed that we are fine tuning. So that's one option that we have worked on. So maybe an email to me and I'll put them in contact with my director to get more details.
Okay. And when fermenting the beans, some farmers dry the beans before putting them in the fermentation box. Does this save time on drying, or why do
No,
they do that?
no. So the pre-treatment of drying beans before putting it in the fermentation is actually specific to, in many instances, to CCN 51, where there's a lot of pulp, and the excessive pulp in the traditional fermentation approach to this variety gave very poor flavor development. And they found with with the pre-drying where the the beans were dried for like 2 or 3 days before starting fermentation, the 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 fermentation and flavor development occurred at a more appropriate rate for this variety that reduce the levels of negative flavors that were present compared to the traditional fermentation that was used for that. So pre-treatment is a function of the amount of pulp and optimizing it to give you a better fermentation. So I know in Malaysia, sometimes they would have employed pressing of beans to remove some of the pulp. Now, if you remove too much pulp, of course, you have a very you have less sugars to start off the fermentation process. So you might have a slower or longer lag in the yeast-based fermentation. So you have less alcohol for the acetic acid stage of fermentation. So it's all connected again. So any pre-treatment has to work for the variety that it is being tailored for.
Okay. When fermenting in the box, you said the height matters. So should they use instead of this, the cubicle box, do they use a longer box?
Linda, it's all about surface area to volume ratio, right? So if you have a small quantity of beans and you put it flat on a tray, you have more exposed surface area compared to the volume. So you actually lose more heat. So tray fermentations with one tray occur at a very rapid rate, and you do not have optimal flavor development. So if you have a small volume of beans and you have to ferment this, then you want to have some vertical element to how that mass is structured as opposed to spreading it horizontally. If, however, you have a large enough volume of beans where you have an optimal surface area to volume ratio, then it's fine. So it's not always necessary to have a cylindrical column for fermentation if you have large volumes. That vertical element or consideration is really relevant only even if you have small volumes of beans.
Okay. What's the relationship between the pod storage time before starting the fermentation and the quality and flavor of the bean that comes?
Okay, so there's some work that was done on this, and we found that now when you store pods, the pods are actively respiring, so they're losing moisture, so the pulp is drying out. In Malaysia, they did some studies where they found that they stored pods up to 6 days, and after 6 days pod storage, then they got noticeable changes in flavor. You got less acidity because you had less pulp moisture content. You probably got more sugar in the pulp. Um, Pod storage up to 3 days, 4 days does not have a negative impact on fermentation, but it does have an impact on how turgid or hard the pod wall is for opening the pod. In that once the pod starts to respire, the pod wall becomes more flaccid or less firm, so it's harder to open that pod. Also, if you have any disease spots on the pod, or if that pod was cracked, the pod husk was cracked, not open, but cracked, you also have oxygen going in, so fermentation could start as well. So the idea is to have as least storage time as possible. It's often not practically feasible to harvest and crack and put the ferment on the same day, but optimally 3 days pod storage should be okay. Once you do more than that, then you have risks of other things happening.
Okay, what is the genotype flavor effect when cocoa is planted in between coconut trees as intercrop?
Yeah, I have no evidence to show that if I plant mangoes between my cocoa trees, that the cocoa beans would taste like mangoes. Similarly, I don't have no evidence to show that that there's a link between planting cocoa trees with coconuts and I get coconut flavor the cocoa beans. I did part of my PhD was looking at pollen donor effect on flavor. That is the male or the pollen going onto the tree, and there's no significant pollen donor effect on flavor, but the maternal effect was stronger.
Okay.
And even between different plants, you have different pollination incompatibility systems. Even within varieties, you have pollination incompatibility systems. So you can't get pollination of a coconut flower with cocoa, for example.
Okay, and then there is somebody's asking about coconut, camphor, and even smoky aromas seem to be related to secondary microorganisms development in the beans. What's your opinion on that?
That is correct. So a deep bed drying that would have some sort of secondary mold growth, that it seems to favor the development of those camphor phenolic chemical notes present in the beans.
Okay, right now it's 12 o'clock. We have a few more questions, or you want to just answer them and then we can post it later, or what do you think?
I think I don't mind answering the questions if everyone's still interested in staying on. So that's okay for me.
Okay. Then just a few more questions, maybe another 5 minutes.
Okay.
Because sometimes for some people it's almost like a midnight for them.
Okay. All right.
Okay. So the drying, is it a good idea to leave the beans outside, or do you have to cover them in the night?
When I said resting, so where you have that sort of the tempering stage of drying, right? You leave the beans in whatever mode it's drying in. So if you have an open floor drying system that's covered at night, whether it's a roof that slides over it or whatever, you would leave it as is. The whole idea is to prevent rehumidification of the beans from external moisture. What you want is to have the opportunity where the beans end the day being dried, and then migration of the moisture from inside of the bean out to facilitate the drying process of that bean itself. If you have a situation where during the night you have rehumidification of the beans externally from an external source, because the beans are not protected properly, then you prolong the drying, the drying process, and you can also get secondary mold growth. So you have to protect the beans from rehumidification, allowing for the migration of the moisture from the beans from inside out.
The other one is fermentation. So primarily it decided by temperature and pH.
No, well, okay. So the end of fermentation, as I said before, you can use different cues. You can use smell, you can use sight in terms of cutting the beans, and you can, if it's possible for you to have the ability to measure temperature and pH, that's great. The more things you have, then you can correlating. So it may not be possible to measure temperature or pH all the time, but if you can associate those or the optimal levels of those measurements to easier measurable things such as smell or how the beans look when you cut them during fermentation, then you have a more rapid assessment. So temperature and pH, yes, but also smell and how the beans look.
Okay, is it possible to ferment different variety of cocoa pods in the same box in the same time?
Yeah, so it's something that we're working on right now, where that's that same divided box that I showed you, we are making some changes to it where we can put different types or different varieties of cocoa in that box, where they remain discrete, but they function as a uniform mass. And it has to do with the conductivity of heat throughout the matrix, whilst keeping them discrete. So it's something that we are working on right now. So probably the next time I talk to you, I can present some results on that.
Is it okay to use insulated foam or plastic boxes as they are easily maintained? Yeah,
we have done a lot of work in using styrofoam boxes. We're trying to do work now where we're moving away from styrofoam boxes because styrofoam is not environmentally sustainable, and many countries are banning styrofoam. So we are trying to find ways to insulate the fermentation mass in a more sustainable way. So if you look at your metal water bottles like this, the insulator in this is not the metal, but it's the air between the metal. Styrofoam is almost 100% air. It's 90-something% air, right? So air is the insulator, really. So it's really looking at finding an environmentally sustainable insulator, and we're looking at seeing how something that has a lot of air in it or air itself can serve as an insulator.
Okay. Somebody is using a neem leaves to protect the dried beans. Is it okay?
Yeah, that's fine. Once it doesn't impart any flavors or flavor notes to the beans in storage. I use bay leaves in my pantry to protect my kidney beans and black eyed peas and so on from moths. So yeah, so those things work once it does not impart an off flavor or taint to the product. Natural is good. I'm all for being organic and natural.
Okay. So the other one is using using boxes to ferment beans. They don't have it in Ghana, so how they can construct? There's a farmer from Ghana.
So I've been to Ghana a number of times, and I know for a fact at the Cocoa Research Institute of Ghana in New Tafo, they have a fermentation box cascade system as a demonstration for farmers to see and learn how to construct fermentation boxes. So I would suggest that they contact the Cocoa Research Institute of Ghana to get more information on what types of woods are available locally that can be used and so on for that.
Okay, this is a question on halal. Somebody is using a food grade drum fermentation for their halal protocol and post harvest. So how do you compare based on your research on wooden box? We use the same method on standard fermentation, but we change only the materials to avoid flavor contamination during the fermentation.
So in the Dominican Republic, they actually have a range of fermentation systems. One of them is fermentation in plastic boxes, plastic drums. And you do in fact get a difference in flavor profile and microbial succession with the plastic drums compared to the wooden systems. They say that the plastic systems tend to give a slightly lower flavor development. I haven't tested it myself, but I think that's what happens in the Dominican Republic. But it's all about what the market wants. So this company that operates a very large fermentation system, they offer many different types of fermentation: plastic drums, wooden boxes turned once, twice, 3 times, 4 times. It's all about what the client wants in terms of the flavor profile that works for them.
I think that's all the questions they have.
Great, great.
But Umaharan is on the line. Should we just ask him to if he wants to say a couple of things?
Prof. Umar, sure, I have no problem. Yeah, if he wants to say a few words, that's great. That's excellent.
We'll make him co-host or
Okay.
yeah, enable his.
So, Prof. Umar, I would like to invite you to say a few words, if you want, please.
I think we have. Teresa, can you unmute Dr. Umaharan? 1 second. I think we have to unmute him, I think.
Okay. I'm actually not seeing him on my list of.
He was there. Okay. I think maybe he's not here. Okay. Anyway, I just want to thank you all for staying late. And I think it's getting late, so maybe we will just get his written communication. And thank you, Dr. Darin, for a very useful and informative session. I think, we want to invite all of you for our next webinar. Teresa, you want to tell them a little bit about the December 26th event?
Okay.
December 26th event.
Oh, yes, sure, yes. We're just having a 1-hour get together on December 26th for chocolate makers, farmers, everybody to come together just for a little time to relax and do a little bit of meet and greet, get to know each other, kind of blow off steam from the holiday season. So it's just an informal gathering. And forgive me, I don't have the time handy, but we're promoting in our social media and through our newsletter. So we hope to see everybody there. We're asking you to come with a smile and have your favorite cocoa inspired drink in hand. So thank you.
So thank you all for your time and happy holidays and happy New Year.
Right. And from my end, thank you all very much for staying. I hope that I was able to achieve the objective that I started off this talk with. And I would, from my end, I would like all of you to have a safe holiday season. Stay safe. Enjoy it with your friends and family. And I wish you all the best. And keep doing what you do with the passion that you have. So from my end, best wishes from myself and the Cocoa Research Center.
Thank you all.